Hi everyone, this is SachinNair here with another video. And in this video, we will build upon the prior one where you perform the activity of sketching a simplified mammalian fear circuit. We will focus on components in the fear circuit, looking at more details in the cellular and behavioral levels. So in this example of Pavlovian auditory fear conditioning, a rodent is conditioned to associate tone with a mild foot shock, as we saw in the previous video. So when rodents hear a tone and are exposed to a shock, they learn that tone will predict shock. Rodents will freeze when the tone is played as a fear response to anticipation of the shock. So let us take a look at this bottom figure. So the y-axis of the figure represents the number of conditioned responses, and in this, it is the observable responses are freezing, and the x-axis represents different phases of the trial, and each dot represents a trial. So let us first take a look at the conditioning phase. So as we can see, as time passes, the rat has a higher number of conditioned responses and therefore is freezing more after hearing the tone. This is intuitive as the rat learns to associate the tone with the shock and therefore begins to freeze when it hears the tone in anticipation of the shock. Now let us discuss extinction. So extinction occurs when deconditioning occurs. So in this example, tone is played without shock in the extinction trials after the rat has been conditioned to associate it with shock, and the rat gradually learns to not associate the two. And there are different phases of extinction, as shown here by acquisition, consolidation, and retrieval. And these terms can be a little confusing, but what is important to understand is that extinction is actually creating a memory of its own and not erasing a previous memory. So let me repeat that again. Extinction is actually learning a new memory of extinction and is not erasing the previous memory that happened during conditioning. So during this first phase of extinction, as seen by this acquisition, there's actually acquisition of extinction. So really, the extinction memory is being acquired. And as we can see, when looking at these initial trials, the rats gradually learn to not that the tone does not predict shock. And so they gradually spend less time freezing. And at the very end of acquisition, we can see that the number of conditioned responses has reached 0. Now, the next phase of extinction is consolidation. And this is just like it sounds when the memory is consolidated. So we can think of that as the memory becomes stronger. So for example, when as humans, we study or learn something in the day, when we sleep at night, it can be further stored into long-term memory. Similarly, in rats, there is a phase of consolidation when the rats sleep after the trials. During the next day, when the tone is played, the animal is able to retrieve the extinction memory, and it is still there or is strengthened. Now, this phase of retrieving extinction memory can be divided into good extinction retrieval and poor extinction retrieval. So good extinction retrieval means that the rat is able to retrieve the extinction memory well. So it has learned to not associate the tone and shock, and it can retrieve that well later on. Whereas poor extinction retrieval means that the rat is not able to retrieve the memory, the extinction memory well, and therefore has not learned to dissociate the two, both tone and shock, and is still pairing tone with shock. Now, there are different causes of poor extinction retrieval, in a normal animal, as have been demonstrated by behaviorists. So spontaneous recovery is over the passage of time, fear can actually come back, meaning that the extinction memory has been forgotten. And then there is something called renewal, which is if a rodent or animal is put back into the context similar to where it was shocked, fear can be renewed. And so therefore, that's why it's called renewal. So it is remembering similar circumstances and contexts, and then the fear memory comes back. And finally, if the animal receives a shock at random or randomly at a later time, the original fear can come back, and this is called reinstatement. And so these interesting findings, again, suggest that the process of extinction is not actually erasing fear, but creating a new memory, which is this extinction memory. And this phenomenon of extinction memory is established in the literature, but the details of how both fear and extinction memories are stored in the brain are still not very clear. Now, these phenomena we discussed so far of spontaneous recovery, renewal, and reinstatement are all true for normal animals. Now, there's a fourth state or a fourth cause that we see right here, which is pathology. And that just means it's a disease state. So something is going wrong. And this has been shown to result in poor retrieval of extinction memory. And this has strong implications in medicine and psychiatry regarding what is going on at the circuit level. And if that can be understood well, this can help with possible treatment of anxiety disorders, trauma, or PTSD. And this is what researchers are addressing presently using a combination of in vitro, in vivo, and computational models. So understanding the fear circuit, including learning of both fear and extinction, may help when dealing with patients with anxiety disorders, including, as I mentioned, PTSD, generalized anxiety disorder, any trauma-related condition, phobias, and understanding how extinction is learned and its neuronal and circuit correlates will have huge clinical implications. So we will discuss a computational model in a later video that captures all these conditions and has direct relevance to them. So in this slide, there is a diagram of the experiment showing the basic pathways and anatomy. And as we can see here, there is a conditioned stimulus, which is a tone, which provides sound waves that are converted into neuronal impulses by the cochlear neuron. And that sends a signal to a neuron in the auditory thalamus, which can then send a signal to the lateral amygdala or the auditory cortex, which can also then send a signal to the lateral amygdala. Now, as we can see on the other side, there's the unconditioned stimulus, which is the foot shock. And this is a stimulus that sends pain signaling from this shock to the somatosensory thalamus. And this can go to either the somatosensory cortex or the lateral amygdala. But it's important to note here that both of these converge at the lateral amygdala. And the lateral amygdala then projects to deeper brain structures that causes a behavioral response. So with the rats, for example, we saw freezing after there was tone and shock combined. And you can also see other stressful responses, such as an increase in blood pressure or releasing some type of stress hormones. And so now we will look at the tone pathway, but an actual circuit diagram of what we just saw. So here's the circuit diagram, and we had looked at this earlier in the previous video, but for brief review, there is a tone that is a sound wave that vibrates at a certain frequency. And that sound wave is picked up by sensory neurons in the cochlea of the mammalian ear and converted into electrical neuronal spikes. And so the sensory neurons can be represented as the yellow triangles, which are just cell bodies of these three different neurons in the pathway. Now, a dendrite is the receiver of each cell body, and so it receives the signal from the previous neuron and then goes through the cell body and sends a signal through a structure called an axon. So as we can see here, this excitatory signal, so from the cochlear neuron, then has a signal, so then goes to this primary neuron, so this neuron in the ear, that will send a signal via an axon to the dendrite of a neuron that's located in the thalamus, in the auditory thalamus. And that travels from this dendrite to this cell body to the axon that comes off this neuron, which then goes to the amygdala and goes to the dendrite here in the lateral amygdala of this neuron. So each of these three neurons have dendrites, cell bodies, and then axons. Now, you may recall that neurons communicate via synapses. So axons from the presynaptic neuron arborize, and the tips of the arbors form synapses onto dendrites of the postsynaptic neuron. So neurotransmitters are released from the presynaptic terminal, and these go and bind onto the postsynaptic neuron, as we can see in this diagram right here. And so neurotransmitter binding open the postsynaptic receptors and then allow for charged ions to flow into the postsynaptic neuron and depolarize it. And this is how electrical impulses are transmitted from the pre to postsynaptic neuron. Now let's look at the shock pathway. And this schematic and circuit diagram is very similar to the tone pathway. So it shows the nociceptive foot neuron that converts this foot shock to spikes and transmits this signal from the foot neuron to a neuron in the thalamus. And that neuron in the thalamus then travels to the amygdala. And similarly here, you can see the actual cell bodies. And so this cell body sends a signal to via an axon. And here is the synapse in the thalamus, where there is a presynaptic axon here, and then postsynaptic dendrite. And then this dendrite, remember, is the receiver that receives the signal. And then signal travels down this cell body, and this cell body sends the next signal via this axon to the amygdala. And here's another synapse, presynaptic, which is represented by here, and then postsynaptic dendrite in the amygdala. And then this signal travels to the amygdalar neuron. So putting both pathways together. So imagine tone and shock pathways are functional at the same time while looking at this circuit diagram. So the Ledoux lab at NYU found that both pathways can converge onto the same amygdalar neuron. Now, this is an important discovery. So if the tone pathway is active without the shock present, the amygdala neuron may not fire much. Recall the habituation phase when the firing rate is about 0.5 hertz. This is because the synapse from the tone thalamus to the amygdala neuron is not very strong. So from the tone thalamus to the amygdala neuron, this synapse here is not very strong. Now, on the other hand, if the shock pathway is active, but without the tone being on, the amygdala neuron will surely fire at a high frequency, which is 7 hertz or more, as you may recall from the previous video, signaling fear, as will happen to humans, or any animal if shocked accidentally. This is because the synapse from the pain thalamus to the amygdala is very strong, as opposed to the auditory thalamus to the amygdala. So this synapse here is very strong, and this synapse is not. Now, here's where it gets interesting. If both tone and shock occur at the same time, the tone thalamic neuron will fire due to tone, and the amygdala neuron will fire high at the same time due to shock. Now, cells that fire together wire together. Now, this phenomenon is called Hebbian pairing, and this will lead to strengthening of the thalamic tone to the amygdala synapse. So this synapse will actually grow stronger. So it is important to note that the firing of the tone thalamic neuron and the amygdala neuron is due to different inputs that happen to be coincident. So that just means they happen to be at the same time. Now, this concept is confusing, but let us look at it in more detail in the next slide. So this slide shows what we touched on in the previous slide of the phenomenon called Hebbian plasticity. And that is that cells that fire together wire together. Note how they fire together due to different inputs. So one is from tone and one is from shock. But how does plasticity occur from this? So a spike in the presynaptic neuron releases glutamate into the synaptic cleft. Now, the glutamate then binds to both AMPA receptors and NMDA receptors located on the postsynaptic cell. So AMPA receptors are ligand-gated ion channels, and in this case, the ligand is glutamate, while NMDA receptors are ligand and voltage-gated. So AMPA receptors, when bound at any voltage, will open to let cations flow into the postsynapse and depolarize it. NMDA receptors, on the other hand, have a magnesium ion blocking the pore of the channel, and therefore do not open even when glutamate is bound, when the magnesium is still present. Now, at around -40 volts, the magnesium stops blocking the pore, and thus NMDA receptor channels can open and lead to further depolarization and permit calcium influx into the cell. AMPA receptors is a mixed cation channel, so they are mixed cation channels and let only sodium and potassium in, while NMDA receptors are also mixed cation channels, but they also let in calcium. So the reversal potential of AMPA and NMDA receptors is 0 millivolts. Now, plasticity or long-term potentiation requires calcium influx into the postsynaptic neuron, which can happen only if the NMDA channels on the postsynaptic side are open. Now, this can happen only if both the presynaptic neuron, which is the tone thalamic neuron, fires, and the postsynaptic neuron, which is the amygdala neuron, fires. Now, this is Hebbian plasticity condition that we just explained when the tone thalamic neuron is fired by the tone and the amygdala neuron is fired via the shock, and that opens the NMDA channels. And the calcium influx into the postsynaptic side of the tone amygdala neuron will result in synaptic growth, which happens in 2 ways. Increased calcium concentration on the postsynaptic side causes increased efficacy of the existing AMPA receptors, indicated by blue in the schematic. And also an insertion of AMPA receptors into the postsynaptic membrane, indicated by an increased number of blue receptors from 3 on the left panel to 5 on the right panel. So let me explain this one more time. So assume that the amygdala neuron is postsynaptic, and the presynaptic neurons are from the tone and shock pathway. So the amygdala neuron is postsynaptic, and the presynaptic neurons are from the tone and shock pathway. The green one on the top is from the shock pathway, which is not plastic, and the green one in the bottom is from the tone pathway, which is plastic. So Hebbian plasticity causes long-term potentiation, causing that synapse to strengthen if tone and shock occur and overlap in time. So look at the difference here. This is initially before the tone and shock fire at the same time, and then this is after. You can see these receptors are upregulated and long-term potentiation has occurred. So ultimately, this tone amygdala synapse from the left to the right has strengthened. That is all for this video, and I look forward to our next video.